On behalf of the Nanion team, I would like to welcome you all to this webinar. It is nice to see so many of you joining for the discussion of this very interesting and hot topic nowadays. So before we start with the webinar, I'd like to give you some general information. Okay, so by default, your video will be off and your audio will be muted. This webinar is being recorded and will be later available on demand. And by participating in this webinar, you consent to this session being recorded. There would be also a joint Q&A session available after both presentations have taken place. For that, we invite you also to write your questions in the chat as they appear in your head. So please write the name of the speaker that you wish to answer to your question. So either Andre or Adriana. And at the end, the moderator will read the questions that you have written in the chat, or you can also raise the hand in the chat, and you can ask or formulate the question yourself if you are brave enough to do that. Yeah, and then at the end, there will be a survey available. Would take only a couple of seconds to answer that, and we would appreciate then your feedback. For the timeline of the webinar, we will start with an introduction to the technology and also the topic that will be given by Dr. George Okayo. Then we will move on with the first presentation of the day from Dr. Andre Bazzone. And last but not least, we will have the presentation from Adriana Bizior. So I think without further ado, we can get started with the webinar. So it is my pleasure then to introduce you to my colleague, Dr. George Okayo. He has been working at Nanion for over 11 years, and he has changed different positions in the company, but now he's working as the Vice President of Nanion Technologies in North America, and he will be the moderator of today's webinar. So when you're ready, George, you can take the floor.
Thank you. Yeah, got that. Yeah, as Gemma mentioned, thank you for the introduction. My name is George Okayo, Vice President, North America. I'll be moderating today's session. The theme of today's webinar is TMEM175 screening on the SURFE²R, and I believe there'll be data on the SURFE² as well. In essence, this webinar focuses on accessing intracellular ion channels and transporters in research and in drug screening. There are so many different types of membrane proteins in lysosomes, cation, anion transporters, proton pumps, ion channels, and many other integral and peripheral membrane proteins. That being said, defect in these membrane proteins leads to impaired lysosomal function and is associated with numerous diseases. TMEM175 is one of those lysosomal ion channels and has emerged as a potential therapeutic target. In general, TMEM175 is a constitutively active potassium and proton leak channel involved in maintaining lysosomal integrity by regulating pH, membrane potential, and autophagy. But that being said, there are many challenges encountered when targeting organelle channels or transporters, for reasons mostly because most have unknown functions, their solution environments not very well understood. Accessibility, of course, is a problem. The organelles tend to be small. Direct recordings are difficult, certainly because organelle membranes are intercellular. And so considering all these challenges, it's generally difficult to access them. But that being said, there are two platforms from Nanion Technologies that have been used for study of TMEM175, automated patch clamp and solid supported membrane-based electrophysiology. But that being said, before I get into anything else, I'd just like to introduce the company for those in the audience who may not be familiar with Nanion Technologies. Nanion is a Munich-based biotech company founded more than 20 years ago at this point, and has grown to be one of the leaders in providing automated patch clamp systems. But over the years, we've grown to not only provide automated patch clamp systems, but also innovate in systems for artificial bilayer lipid membranes recordings, systems that are used for cell analytics, such as those used for label-free real-time cellular readouts, like impedance systems, extracellular electrical recordings, contractile force measurements, which would be the flex site, as well as systems used in solid-supported membrane-based electrophysiology. Now, considering that, clearly, we aspire to be that one-stop shop for membrane science. So if you have any project whatsoever related to membrane recordings, we likely have a solution for your applications. Therefore, please let us know how we can partner with you in accelerating your research using one of our technologies. But for now, I'd briefly like to touch on those systems that were used for generation of TMEM175 data, which would be our automated patch clamp system, SURFE² 384, as well as the SURFE² technologies. We have pretty much 4 automated patch clamp systems, the PortaPatch family of instruments that record one cell at a time, but have add-ons for external perfusion, internal perfusion, their capabilities of temperature control, and so on. And then we have the patch liner that can perform 4 or 8 cells recorded at a single time, which gives about 600 data points per day, translating to anywhere between 75 and 300 compounds per day. The patch liner is pretty flexible and allows for use with add-ons such as temperature control and dynamic clamp. And then, of course, we have our high throughput SURFE² 384 that can record up to 20,000 data points per day, with other capabilities and features such as rapid solution exchange, making the SURFE² 384 suitable for working with fast sensitizing ion channels. We have temperature control, which includes heating and active cooling, internal perfusion capabilities, and current clamp, therefore working with the action potential measurements. And of course, high throughput concentration response curve generation. But that being said, the SURFE² is not only usable for screening of large compound libraries. Using the 32-well mode of operation, SURFE² can actually become like a lower throughput system that would be usable for smaller screens and for assay development. Therefore, allowing the system to only use a fraction of the chip and the pipetting head in multiples of 32. Talking about the SURFE², there are terminologies that you'll hear about, or acronyms that you'll keep hearing about today. SSM stands for solid supported membrane. And the name SURFE² itself comes from surface electrogenic event reader. There are 2 of these systems, the SURFE-N1 and SURFE-96 SE, that at this point are used in academic labs, pharmaceutical companies, CROs, and many different settings where similar type recordings are performed. I'd like to briefly just touch on the measurement principle, although Andre will go deeper into discussing more of this. The point I'd just like to make here is that the SURFE², unlike automated patch clamp systems, does not do voltage clamping. Membrane transport is triggered by the application of a chemical gradient, which could be a substrate, an ionic gradient, or pH gradient. In general, the workflow involves registering basal conditions and then perfusion of the substrate, which, because of the capacitive coupling between the vesicles containing the transporter or protein of interest and the sensor, which has a solid supported membrane, we charge the membranes and an on signal can be generated, which is then turned off with the perfusion of a non-activating solution that gives the off signal, and the signal peak and integral are therefore used for the analysis. There are many features that I'd like to point out here, but of major importance would be that the recordings are stable for hours on the SURFE²R. It's ideal for assay development and for academic research, but it's a single well recording system. Sensors look like what you have on the screen here. The SURFE² SE, on the other hand, is higher throughput, recording parallel 96 from 96 wells, and it's very low sample consumption, up to you can use 0.1 micrograms of total proteins per well. One of the really cool things about the SURFE² is that membranes can be stored at -80, so there's no need for continued cell culture throughout your screening campaign. And with that, I'd like to introduce our speakers, Andrew Bazzone, who will be talking about functional investigations of the TMEM175 selective for protons and potassium ions. And he'll be followed by Adriana. But for now, Andrew, please.
Thank you, George. Let me share my screen. So here is actually the contents of my talk. It will be divided in 3 sections. The first section will be basic concepts about the SURFE² technology and SSM-based electrophysiology in general. I will talk about advantages and also limitations of that technique. Then I will switch to the TMEM175 data we collected on different SURFE² instruments. And in the last 2 slides, I will highlight some results that indicate that the target membrane, where TMEM175 sits in, actually affects the compound pharmacology that is found. So starting with the basic concepts. As George already introduced, the SURFE² instruments are essentially pipetting robots. They will inject solutions containing the substrate of the target protein, thereby introducing a substrate gradient that acts as a driving force for the transporter or the channel, and at the same time as a stimulus for the recording. So once you apply a substrate, the substrate gradient will trigger the flux of charge across the sample membrane. And then the sample membrane will be charged. There will be a voltage generation. And that voltage generation acts as a counterforce for the substrate gradient, which is applied. And thereby the current you will record will decay within a few hundred milliseconds. And what we typically use as a readout is the peak, where no significant voltage was generated due to transport. So that will essentially reflect the steady state transport rate under the given conditions, which means the substrate gradient you applied. So let's have a look here at the list on the left. This will sum up some key features. First of all, the SURFE² or SSM-based electrophysiology in general will allow for real-time recording. So you will observe time-resolved currents. And from these currents, you can even derive kinetic rate constants if you are measuring not only transport, but some other conformational transitions within the protein that are triggered by the substrate binding. The SURFE is HDS compatible, as George already mentioned. We have the SURFE² SE, which is able to measure 96 sensors at the same time. A very big and interesting aspect is that you can access any kind of membrane. You usually purify membranes of a specific source. You can purify organelles and attach it to the sensor. You can purify protein, reconstitute them into proteoliposomes and attach them to the sensor. We even did some recordings on whole cells, so there is essentially no big limitation regarding the source. Then you have a very high sensitivity. This comes from the pretty big sensor surface of about 3 millimeter in diameter, which allows about 1,000 times signal amplification compared to patch clamp, which is limited by the surface of the living cell. And this allows to record also slow transporters, which are hardly accessible via conventional electrophysiology. And the sensitivity is even so high that you can measure conformational transitions within a transporter triggered by substrate binding. And last but not least, talking about reliability, success rates are pretty high. You are not limited by gigaseals or the variability of living cells. So actually, yeah, so average success rate, I think, is 98%. You can do sequential measurements using the same sensor because the attachment is pretty stable. So you can perform like 20 measurements in a row, and you will get very reproducible currents over that time period. And last but not least, the sensor-to-sensor variation is also quite low, so you can compare currents also across sensors. Coming to the key limitations, and there are two pretty big ones. The first one is that there is no direct voltage control, and that sets the technology really as a complementary one to conventional electrophysiology. That's just because you essentially measure the change in voltage once you apply the substrate gradient across the membrane. So if you would clamp the voltage, there will be just a baseline. And then as an advantage, there are no leak currents, essentially. But on the other hand, instead of leak currents, you will observe artifact currents coming from ion membrane interactions. So if you perform a sodium concentration jump or potassium concentration jump, these will interact with the lipid head groups that are partly negatively charged. And then you will get some capacitive currents as well. And that's why you need negative controls to be performed. Negative controls don't have the transporter of interest, and you only see the membrane ion interaction, and you can subtract these currents from your sample currents, essentially. Here is, again, a slide showing how a measurement trace looks like. George already showed it just briefly again. So you essentially need two solutions at least. One non-activating solution, you record the baseline, then you switch to the activating solution, which contains the substrate of your transporter. You measure a peak current because the initial transport rate is highest once you apply the full gradient. Then actually the chemical gradient decreases over time while the potential across the sample membrane increases, and that will lead to a current decay. Then you wash off the substrate again, you measure the efflux, and well, the whole thing takes 3 seconds. After that, the initial conditions are restored, and you can measure with a different condition, with different substrate concentration, applying inhibitors, enhancers, and so forth. Here are the two devices we have. This is the SURFE²R, a single well device where you can record up to 150 data points per day. And then we have the 96 well plate based device, the SURFE² 96SE, where you can record up to 10,000 data points per day. Of course, you have, given the different hardware, also differences in the liquid handling and in the software. In general, the SURFE²R is an easy to learn instrument. It is very flexible for assay development, while the SURFE² SE comes also with a very powerful automated data analysis software and is very suitable for secondary screening, for example. So this was about the background about the technology. Now we are coming to our results we got from TMEM175. We actually applied both SURFE² instruments. I will note which data sets are from which instrument. Here on the left-hand side, you see full traces now recorded on the SURFE²R. In blue, it's TMEM175, and in red, it's control lysosomes without overexpressing TMEM175. Our standard conditions are essentially a 140 millimolar sodium chloride background, and then we switch from, we remove 50 millimolar sodium chloride and replace it by 50 millimolar potassium chloride in the activating solution. So we essentially see potassium flux in this trace here. And potassium is provided here at 1.1 seconds. So you see about 12 nanoamps potassium flux signal, while you see -1 nanoamp in the controls. And what we actually used are purified lysosomes for both sample and control. So we have HEC293 cell lines overexpressing and not overexpressing TMEM175, and we purified lysosomes from this cell line via a sucrose gradient ultracentrifugation. So the first thing we did is looking at the ion selectivity, and we did this by performing 50 millimolar of cation concentration jumps against 50 millimolar NMDG. So essentially in the non-activating solution, we had 50 millimolar NMDG, and in the activating solution, we have 50 millimolar of the different cations listed here. And we did the same experiment on the control and on the sample. And here the bars actually reflect the peak currents subtracted. So essentially TMEM175 peak current minus control peak current. And from the currents, you can see that TMEM in our assay conducts potassium, rubidium, and cesium, but not really lithium, sodium, and choline. We also tested 4AP. So 4AP is an inhibitor, a known inhibitor pore blocker for TMEM175. And we repeated the measurements, both again, control and TMEM sample, in presence of 4AP, and the corrected signal is here in orange, and you can see that all fluxes, potassium, rubidium, and cesium are fully blocked in presence of 4AP. We then wanted to look into the concentration dependence for the potassium flux, and here we provided concentration jumps from 1 millimolar to 300 millimolar. This experiment was actually carried out with the SURFE² SE. So we used one column of sensors, so 8 sensors for each concentrations. We performed the experiments on the sample again and on the control. Again, for the control, you see negative currents, while on the sample, you see positive currents reflecting the potassium flux. And when you subtract the currents you observe with the control from the TMEM currents, you get a curve that looks like here. It looks kind of hyperbolic, but in fact, if you look at the first few data points at low potassium concentrations and the last 4 data points at higher potassium concentrations, it's very nicely fittable using a linear regression. And from the linear regression, you can actually achieve the the permeability for potassium, which seems to be concentration dependent. So we keep that in mind. There are two permeabilities for potassium depending on the potassium concentration range. We then wanted to know something about the pH dependence. As you all know, TMEM175 not only conducts potassium, rubidium, and cesium, but also protons. We started out to investigate the effect of pH on the potassium flux through TMEM. In the upper figure here, you see the typical measurement of potassium flux, so 50 millimolar sodium against 50 millimolar potassium jumps as a driving force. And we did that experiment using different pH values from 3 to 10. And as you can see, there is a pH optimum close to 8. And then you see downregulation in the acidic pH range and in the alkaline pH range. The acidic pH range, actually, the downregulation is described by two different pK values. So you see like 60–70% activity loss with a pK of 7. And then again, the rest of the activity is lost with a pK of 4.5. These pH experiments were performed using the same pH values inside the lysosomes and outside the lysosomes. So in the next step, we wanted to figure out where this downregulation comes from. Is it the internal or the external pH? And therefore, we carried out a similar experiment, but we kept the external pH to 7.5, and we only changed the internal pH. And here you can see the curve that comes out from this type of experiment. And as you can see, if we acidify the lysosomal pH, actually the signal increases, the activity of TMEM175 increases, which makes sense, right? Because TMEM175 is a lysosomal cation channel, and it has naturally acidic pH environment from the internal side. But if you go lower in pH, you see the signal loss again. We think this signal loss is the same signal loss as we see at symmetrical pH, just due to competition with protons. So since you have much more protons at acidic pH values, protons go through the channel as well, and that will block the potassium flux, which we measure here. So as a conclusion, this downregulation we measured at symmetrical pH values must be due to changes in the cytosolic pH environment. And we think this is a kind of a mechanism to stop further acidification of the cytosol once the cytosol tends to acidify. So this was the effect of pH on potassium flux. Now we wanted to measure the proton flux directly through TMEM175, and this is done by pH jumps. So we did two different types of experiments. The first experiment was equilibrating the pH to 7.6 and then acidifying from the external side, essentially measuring proton influx driven by the pH gradient alone. And you can see here, these are the currents we measure with the different delta pHs. Of course, the higher the pH gradient, the bigger the current. And with the highest pH gradient, 3.0, which is the orange curve here, we also added 10 millimolar 4AP to block proton flux through TMEM, and we get this black current here. And what is remaining here is the artifact resulting from proton-lipid interaction. We actually measure exactly the same current when we measure control lysosomes without 4AP. So that's all nice. We actually measured the control as well and subtracted the currents. And here are the artifact-corrected currents. And plotted against the proton concentration. Instead of delta pH, it's delta proton concentration. And here, if we fit linearly, we can get permeability for the protons. Here you can think of why is this here not linearly increasing at higher proton concentration jump, and the answer is that you actually see downregulation at acidic pH values. So if you acidify much more beyond neutral conditions, then you see that downregulation effect, and you actually don't see a linear increase. So we just used the first few data points to derive the permeability. We did the same thing with efflux. So we have as initial conditions the pH equilibrated to 4.6, so very acidic, actually resembling natural lysosomal pH. And then we provided more alkaline pH values from the external side and measure proton efflux. And here, essentially, you see the same thing, only if you jump to very alkaline or close to neutral pH in the outside, then you will see a linear dependence, and you can get the permeability for protons, which is essentially the same as for influx. So there is no different permeability for influx and efflux in our assays. And one very nice result is that if you take the ratio of the permeability for protons and the permeability for potassium, you will get a number which is 48,000, meaning proton permeability is 48,000 higher compared to potassium permeability. And this matches very nicely to the permeability ratio found by Hu et al. And he applied patch clamp to obtain this ratio, and this validates the SURFE² or SSM-based electrophysiology approach very nicely. We then also measured some compounds, some tool compounds. I will just go very briefly through that. A typical workflow sequence here is that we do 3 times activation of TMEM175 using 50 millimolar potassium. This is essentially the reference current we have. And then we incubate, we provide the compound, we incubate for 5 to 15 minutes, depending what kind of compound we have. You need larger incubation time if compounds are hydrophobic and need to intercalate into the membrane, so that takes a little more time. And then we repeat the activation using 50 millimolar potassium in presence of compound. Here you can see the result for an enhancer, and here the DMSO control. And after that, you can wash out the compound again. And you can see that for some cases, in some cases, for some compounds, you can wash it off. For some others, you can't wash it off. If they are very sticky, hydrophobic, they just stay in the membrane and you won't see the washout. We actually measured inhibitors, zinc and 4AP, and derived IC50 values. And we also measured enhancers, DCPIB on the left and arachidonic acid on the right. So this is the last section of my presentation. And here I actually only want to provide a kind of teaser. It seems, or some results we found indicate that the target membrane is very essential and affects the compound pharmacology when assaying TMEM175. So first of all, I would like to go back to the assay conditions we have with the SURFE² instruments. I told you we use purified lysosomes, but that's not the full story. So yes, we purified lysosomes. We used two different techniques that have been published by Schulz et al. And by Jin et al. 2008 and 2017. But we also checked for lysosomal and plasma membrane markers of these purified lysosomal samples. And as you can see here, with both techniques, we get LAMP1 concentration, which is the lysosomal marker. So there are a decent amount of lysosomes there. But we also find sodium-potassium ATPase, which means there is some contamination with plasma membrane vesicles. So essentially, since for the SURFE² we measure macroscopic currents, we will have or likely have a mixture of lysosomes or TMEM characteristic in lysosomes and TMEM characteristic in plasma membrane vesicles. Why do I tell you that? We did a study together with Cerevel as a collaborator, looking at 8 enhancers, potential enhancers for TMEM175. And we performed SURFE² assays, as I just introduced, but we also performed whole cell automated patch clamp using the SynchroPatch, where we actually have only TMEM in plasma membrane. And we also performed manual lysosomal patch clamp, where we have 100% of the TMEM residing in the lysosome. And interestingly, the enhancers we tested can be clustered into 2 groups. Cluster 1 has the enhancers in cluster 1 show very similar potency with all 3 techniques. In the cluster 2, lysosomal patch clamp shows no activity, while whole cell automated patch clamp shows quite large activity. And the SURFE² assay actually is somewhat in between. And that's why I was mentioning that we have here a mixture of plasma membrane vesicles and lysosomes. So that could explain why the SURFE² is basically in the middle of these techniques. And for cluster 3, it's a very similar thing, but here we see high activity in lysosomal patch clamp, essentially no to low activity in whole cell patch clamp, and the SURFE² again is somewhat in between. Why can that be possible? So our first thought was, well, if a protein resides in a membrane and interacts with lipids there that are only available in that target membrane, or also with other proteins that are only available in that target protein, the interaction sites of the compounds will change, right? So that could have a major effect. And luckily, just a few weeks ago, there was a publication showing that the LAMP proteins actually directly interact with TMEM175, and that could explain these changes. So we think that it's really important to look for compound potencies of TMEM175 in its natural environment, which will be the lysosomal environment. So that was it from my side. I just wanted to highlight a few resources. There are 2 nice book chapters from 2017 and 2020 where you can dig into the SURFE² technology in general, and also just released this year a publication which highlights more the data analysis part, also looking into pre-steady state currents, binding currents, and transport currents, and how to distinguish those. And the TMEM175 story is actually currently under review, so we hope that this will be released soon. And if you want to see the SURFE-N1 in action, there is a YouTube video you can access here, where Maria will show you sensor preparation and guide you through some measurements. And now I would like to hand over to Adriana, who will continue with some results on TMEM175 on both SynchroPatch and SURFE²R.
Thank you, Andrea. Can you hear me okay?
Yes.
Just at the beginning, I would like to thank Nanion for organizing this webinar and for inviting SB to give a talk today. So as George and Andrea said, I'm going to be talking about utilizing SURFE² for TMEM175 studies here at SB. Sorry, I think my laptop was lagging. So I think it's needless to introduce TMEM175. It is an exciting new target with therapeutic potential. And here at SB, our aim is to develop robust reagents and screening cascades to facilitate TMEM175 drug discovery screening. And for that, we have a few cell lines prepared with a wild type TMEM175, human one, as a cell line, as well as two variants, loss of function and gain of function variant, and on the way, mouse and rat orthologs of TMEM. But before I hop on the SURFE² train, I would like to briefly talk you through our journey with TMEM175 using electrophysiology platforms. So our aim is to develop a robust cesium-based automated electrophysiology assay, and we achieved that with wild-type TMEM175 being expressed in inducible hex cell line. And as you can see, the TMEM175 in inducible cell line, we pretty effectively created a baseline, which then could be inhibited by the 4AP in a concentration-dependent manner, and then it could be activated by zinc chloride, again to be blocked by 4AP. This was validated in the cell line with not overexpressed TMEM175, and we can see any appreciable current apart on the untransfected cell line, which was good. And using automated patch clamp, we identified novel TMEM175 modulators. So we screened about 20,000 compounds, and using this assay, we were capable of identifying both activators and inhibitors. And fluorescence-based assay showed a great correlation with our APC assay, and thanks to it, we successfully measured activity of hit compounds and reference compounds with those two platforms as cross-referencing each other. So here's just a table with chosen hit compounds, TMEM activators in our fluorescence and electrophysiology and their EC values for them. But of course, we know, Andre mentioned that as well, that TMEM175 is not just a potassium, cesium, or rubidium channel, it's a proton leak channel in lysosomes. It's highly proton selective, and it's gated by luminal protons, and mediates lysosomal proton efflux. So here at SB, we wanted to develop this assay to measure the protons using automated patch clamp, and we developed an electrophysiology assay to measure inward proton conductance. And we found this sweet spot, as we call or refer to, at pH 6. So this just represents a pH CRC of TMEM175 and an application of different pH values going from 7.2 to all the way down to 4.5. And hopefully you can see that the lower the pH, the higher the proton conductance. And now what I mean by the sweet spot at pH 6. Well, we know that TMEM is activated by the pH itself, and it's also activated by the DCB-IB compound. So we kind of chose this point based on this data, where activation by pH doesn't quite reach the maximum capacity of TMEM itself, and where it's still at the same pH can be activated to its maximum by the DCB-IB. So based on this, we developed an assay, a low pH assay, where we measurable proton conductance stably transitioning from pH 7.2 all the way down to pH 6. And hopefully you can see that as soon as you drop the pH, the amplitude recorded is larger, and it's a pretty stable current, and it is then can be blocked by 4AP. And this block by 4AP is pretty consistent and is concentration dependent as well as visualized on this graph here, but also with EC50 value over here. And a good thing was that no effect on untransfected cells was recorded during those conditions. So we took this assay a little bit further, and as opposed to blocking the signal, we wanted to enhance the proton conductance, and we did that with the use of DCPIB as well as our preferred SB compound. And we found that EC50 values were pretty consistent with the original screening assay. And here's just to represent that DCPIB. It was pretty consistent and concentration dependent when pH 6 was dropped and different concentrations of DCPIB were applied. It was nicely plotted on the graph with a nice EC50 value, and all those currents were blocked by 4AP. Similar situation with our SB compound, also quite a nice concentration response curve. And yeah. But of course, all this work was done on live cells. And while lysosomal patch clamp is possible and we will be working towards it as well, it's quite challenging at the moment and not really cost effective. So other electrophysiology platforms needed to be explored. And that's where solid supported membrane electrophysiology, SSME, comes into play. And of course, I'm not going to be going into any details because we had George and then Andre explaining it very well, all the technology, but let me just briefly recap the advantages of using SSME for those purposes. So no living cells are required. As Andre mentioned, those are just membrane stored at -80, so no continuous culture needs to be performed for those assays. SSME measured transport of charged ions, and it's ideal for studying electrogenic transporters. Its high stability of the sample allows multiple repeat experiments, as Andre said, on SURFE²R, up to 20 measurements are reproducible, and there's no loss in currents in those circumstances. And of course, it's suitable for studying lysosomes, which what we're interested in. So apologies in advance for this quite busy slide, but that's just to recap what Andre was saying, and I'm showing you this just to prove a point of sample validation. Sample of TMEM175 that we prepared here at SB using our own cell line and validating it on SURFE² here in-house as well. So just very briefly about selectivity of TMEM175, not to such a great detail as Andre did, but we do see similar behavior where cesium is a preferred ion here in green over potassium in orange. And also this is just a 50 millimolar of potassium on the graph in this case of the untransfected cell line that we used for preparation of our lysosomes as a negative control. And you can see there's no current recorded or a negative current recorded at those conditions. EC50 values for both potassium and cesium were pretty consistent as well, meaning that the assay was working very well. And then again, just as principle, we tested known blockers and activators on our sample made here in SB, and we did a concentration response curve for AP, and this is just a graph representing what it looks like on SURFE² as well. And we also performed some zinc block as well, which was also pretty successful. Then, of course, we also wanted to test some activators, and the preferred one and known for TMEM175 is DCPIB. And you can see a nice concentration response curve. Again, just a graph representing what it looks like on SURFE² on the currents. And we also chose another activator ML6733, which is a known activator for K2P potassium channels. And while the CRC is looking pretty convincing, it is in a 2-digit micromolar range here, about 15 microliters, but it doesn't fall far from the EC50 values for K2P transporters as well, which range between 20 to 30 micromolar EC50 value. So once we had our sample validated on SURFE²R, our own sample made from our cell line, and we were pretty happy with the results, and they were comparable to what Nanion was seeing as well, we could take our screen, compound screen on SURFE² further. So we found a few candidate compounds activated TMEM175 on purified lysosomes, with an example here on the right. But strikingly, some of the compounds were not activating TMEM175, including our preferred SB compound that we've been using routinely in electrophysiology, as well as on fluorescence assays. So kind of to understand why this could be the case, why we've seen so discrepancies between the compounds and testing them between on electrophysiology platforms and fluorescence, we did some additional tests. And for that, we tested another compound, known compound to be activated TMEM175, which is SC79, along with our SB activator. So SC79 is known to be working, or assumed to be working, on TMEM through AKT pathway, so not directly. So we kind of thought this could also be the case with our SB activator and all other compounds that are not activating TMEM on SURFE²R. So this is just a graph with average peak currents representing different concentration of both activators, and you can hopefully see there's not much happening, not much activation apart from the background noise is going on. And as I said, SC79 works on TMEM through AKT pathways. So we did a fluorescence study where we used thallium as a proxy to measure potassium translocation on live cells. So this is just no compound added, just fluorescence based on only on cells. Then we added our SB compound, and hopefully you can see the activation happening. Same happened with SC79, nice activation comparison to no compound bar. And again, we tested DCP-AB as a kind of positive control too, also seeing a good activation. So SC79 obviously activated TMEM175, and then we wanted to see if it really can be reversed by adding AKT inhibitor. And yes, that was indeed the case. You can see that with addition of inhibitor, the fluorescence dropped almost to reflect the no compound fluorescence. That, however, was not the case with our SB compound or even DCP-AB. What kind of made us believe and assume that it could still be working on TMEM, but through a different pathway, still indirectly, but through a different pathway. And that kind of presents SURFE² as a great cross-reference tool between different platforms. So something that other platforms are not, cannot provide at the moment. And having this in mind, that provides and opens up new and exciting avenues for the drug discovery, where we can at least to some extent say, well, those compounds, not just for TMEM175, but other transporters as well, whether the compound works directly on the transporter or it's working through another pathway. So then, of course, we know that TMEM175 is influenced by the pH, and here's just to reflect 50 millimolar potassium flux at different pH values, and hopefully you can see that with lowering the pH, the potassium translocation drops. So that's something that, of course, Andrew went into more detail, but that's something what we also want to explore here at SB. So having this in mind, we also wanted to see what native environment of the liposome would look like on SURFE²R. So for that, we internally acidified the inside of the liposome, and applied currents, applied swift change of external pH. And with that, we observe that internal acidification potentiates the permeability of TMEM175 to potassium. So here we have the average peak currents adjusted with untransfected currents for the pH 7.2 at 10 millimolar potassium. And nice reproducible currents. And then once we switch to the pH gradient measurements, meaning that we acidified the inside of the liposome to pH 6, and then swiftly applied different pH of 7.2 in this case with 10 millimolar potassium, there was an increase in current, which was pretty consistent. And the percentage increase was roughly about 15%, give or take, pretty consistently across different sensors. Then we wanted to take this even further and see if we can potentially DCPIB. So what we did, we repeated the same experiment, but now with the addition of DCPIB with the EC value obtained with CRC. So hopefully you can see that the gradient measurement without DCPIB is potentiated by DCPIB, and the percentage increase were roughly about 50%, again, quite consistent across different sensors measured on different days as well. And using similar setup, we can also track the proton currents on SURFE² for TMEM175. Again, proton currents were measured by the application of swift exchange of external pH. In this case, in none of the solutions, potassium was present, so it was just application of non-activated solution to adjust the lysosomal pH to wanted pH, in this case, either 6 or 4, and then application of, again, non-activated solution at a pH 7.2. And a larger conductivity was observed when using acidic solution internally. So here we have the H conductance using non-symmetrical pH gradient, pH 6 inside, 7.2 applied externally, in comparison to pH 4.5 and 7.2. You can see a massive increase in the proton conductance. Again, as Andre explained, those currents are adjusted to what we are seeing on untransfected, so this reflects only the translocation of protons via TMEM alone. So now kind of the last thing I want to tell you about is the preliminary data that we got for a loss of function mutant variant of TMEM175. And our initial data shows that variant displays concentration-dependent activation by potassium with EC50 comparable to the wild type. And TMEM loss of function also shows a clear activation by DCPIB. And here's just a couple of concentrations tested at pH 7.2 with 10 millimolar potassium present. And those conditions in this current can be blocked by 4AP as well, as shown in green here. So now just to kind of wrap everything up and summarize, I want to share quite an exciting news as well. The acquisition of SURFE²R96 platform for a high throughput compound screening, not just TMEM, but other transporters, and the fact that we're expanding protein transporter research as well. So here's just the list of neurotransmitter transporters that we have on the shelf right now that are suitable for the SURFE² analysis, but we have another 20 plus targets on the way that are being under development right now, which is quite an exciting news. So again, just to kind of summarize and wrap everything up, our TMEM175 research, we have well-established fluorescence high throughput screening techniques, as well as automated electrophysiology assays for proton and potassium conductance as well. And now we also have SURFE²R, which is quite a powerful tool for cross-reference between those two platforms. We validated the assay for potassium and proton conduction using SURFE²R. And we also now tested loss of function variant, and we have an assay ready for your research needs. We are also acquiring a big SURFE²R, the 96 one, for a large screen compound, with which we will be expanding our protein transporter research too. So again, I would like to thank Nanion for organizing this webinar and for inviting SB Drug Discovery to give a talk. It was a pleasure for me to represent SB Drug Discovery. Of course, we will have a Q&A session just in a minute, but if you forget about anything and you want to get in touch, you can send me an email directly or find an inquiry email on our website. So get in touch, and thank you very much for your attention. Thank you for having me.
Yeah, thank you for that talk. We have a bunch of questions that we will go into right away. First, a few will be to Andre. So Andre, what is the limit of channel protein density in the vesicles, and are channel conductances important for measurements?
Yeah, good question. Channels are in general much faster than transporters. So we don't know. I think no channel has been measured in a reconstituted system where the density is actually known. So obviously the channel density for the TMEM175 assays are more than sufficient. So actually we used 1 to 100 dilutions of the sample we have, which still got yeah, decent signals. So overexpression of TMEM175 is more than sufficient. That's at least what we know.
Okay. And yet another question here. If you are looking at the activity of endogenous transporters, so those that are not overexpressed, what would you use as a negative control to correct for artifacts?
Yeah, native samples are difficult due to that reason. So usually you would prefer to have a knockout as a control, and that's, I think, the only way to control that.
Okay.
But on the other hand, I should add maybe there are assays where we know that the artifacts are not an issue. So artifacts are only an issue with certain types of concentration jumps, like if you have ions or very hydrophobic compounds, and usually you can make good intellectual guesses about if artifacts are an issue in that type of assay or not.
Okay. Another question, on a structural level, I would imagine there are two permeabilities for potassium depending on concentration and the pH value. Is it known if amino acids responsible for permeation are protonated?
I'm not sure about that. I couldn't find any information in literature. I looked for that, but I don't think it's known.
Okay. And is it possible to change the lipid composition of the solid supported, the lipid membrane on the sensor? And would it make sense to do that in the context of TMEM175, considering that it is in an endogenously different lipid composition?
So usually if you want to check the effect on your target protein, then you change the lipid environment of the target membrane, which can be only done directly if you have proteoliposomes, so purified protein, and then reconstituted into an artificial lipid mixture. It wouldn't make sense to change the lipid of the solid supported membrane, because that's just where your sample attaches, right? So we only use that one specific lipid for the SSM surface.
Okay. Yeah, another question here regarding the sensors. Can they be reused for application of other organelles, and which organelles are robustly measured on the system? And a follow-up question to that, how many changes can be made in terms of the different solutions that can be applied?
So to the reusability, we only guarantee a first-time, single-time use of the sensors, but by experience, we know that the sensors for the single well device can be reused like maybe 5 times. You basically recycle the sensors with SDS solution to strip off the sample, and then you can start all over again with a new sample. So maybe 5 times, I would say. Second question was about the different samples or organelles that have been measured. So there are publications who measured mitochondrial membranes, ER membranes, that was mainly SURFE² that have been measured, and I think also synaptic vesicles. So these have been published, but I won't say that there are any limitations as long as you can purify the membranes, you can attach it to the sensor. And regarding the number of solution exchanges, that highly depends on the type of assay and on the type of sample and target. As long as the target protein is stable in the solution environment and the assay conditions are stable, like if you apply gradients, additional ion gradients, those also need to be stable over the membrane. Then you can measure a decent amount of time. I think on average, I'm measuring 20 times per single well sensor, like for the SURFE²R, but you can measure really much more than that. I once did a stability measurement on the SURFE² 96SE on EAAT3, and I measured essentially for 18 hours continuously, and it was just stable during that time. So it's pretty stable. The sensor is pretty stable as long as the protein survives at room temperature. Yeah.
Okay. And one last question from that person asking is it possible that multiple organelles could bind to one chip, therefore affect the readout? And if so, how would you normalize for that?
Yeah, of course, if you have impurities, if you have samples consisting of different membranes, and that's what we realized was the case for the TMEM175. And I should add, we are now really focusing on optimization of the purification technique to really have pure membranes. But if you have impure membranes, then those will contribute to the readout, of course. And there is no clear control for that unless you know how to distinguish those. If you can specifically inhibit one of both, for example, but if you don't know that you can't control for that. Yeah.
Okay. Yet another question. 4AP seems like a planar and electron-rich molecule. So have you noticed any change in sensor parameters, so capacitance or conductance here when using it?
No, that was fine. No changes.
Okay. So there's a question here. Someone asks, can you comment about how you'd set or control the intralysosomal pH that is altered in some of the experiments that you outlined?
So essentially, biological membranes are pretty leaky for protons. And we use that with the SURFE² by changing the pH and then incubating for 3 to 5 minutes. That will equilibrate the internal pH. And the measurement is then started using the making a change of the external pH, and like 700 milliseconds after exchanging the external pH, we will provide the potassium in that case to measure the potassium flux in presence of the gradient. So proton gradients are quite stable within a second or two, but they are very unstable in the minute time range, so that we can re-establish pH gradients during that incubation.
Okay. This one is for Adriana. Could you comment on the difference of effects seen using zinc by APC compared to the SURFE²R, and for the activator and inhibitor effects?
Sorry, do you mean between APC?
So there's a discrepancy, or so let me say differences in the effects when using zinc chloride by APC compared to the SURFE²R.
Yes, so on our hands, and I'm also pretty sure that Nanion saw as well, we see activation. We see activation of TMEM with zinc on synchropatch, and we see inhibition on SURFE²R. I cannot tell you why exactly this is the case. We've discussed it internally. I'm not entirely sure. Other groups as well, other companies also see activation. Yes, activation in electrophysiology. So I'm not entirely sure why this is the case. It could be that it's working on some sort of pathway, and because on SURFE² we're using purified lysosomes, that pathway could be removed and it's working differently on SURFE² than it is in electrophysiology.
Okay, and yeah, for Andre, I don't know if you have any other comments on that discrepancy, I guess also seen in what you showed by zinc.
Yeah, I totally agree with Adriana. I think it has something to do with a different pathway. That's, I mean, for SC79, it's pretty much similar, right? There we don't see any effect on the SURFE² because the protein kinase is missing, and yes. SC79 acts via that protein kinase. And maybe with the zinc, it's a similar thing.
Okay. And for Adriana also, someone mentions here that they didn't understand if you have an explanation for why the EC50 are kind of different between the fluorescence assays and electrophysiology. Could you please elaborate on that?
So again, I'm not a specialist on APC, but I think it could be due to lots of different factors. Perhaps APC is obviously EC50 value is a little bit higher. It could be due to the fact that more cells are patched or are patched in a different way, well to well, so it's taking the average, and perhaps that's why it is slightly higher. But again, I'm not working in electrophysiology myself. I've solely been working with SURFE²R, so I'm happy to answer that question. If the person is happy to send an email, I can direct them to a specific person, but I personally cannot really comment much on this.
Okay. Yet another question here for either of you, either Andre or Adriana. Are the recorded currents from isolated lysosomes generated on intact organelles with the contents retained, or are they sonicated such that they just have the lysosomal membranes without the contents? And are these preparations stable at -80?
So I think we'll have similar answers to that. The sample has to be sonicated before application on the solid surface, and it's due to the fact that when you remove your sample from -80, you'll have all the lipids kind of clumped together. So you want to make sure it's a nice and dispersed solution. So yes, they have to be sonicated before application, and what goes with that, potentially the insights are lost. But I don't know if Andre, do you want to add anything yourself?
Yeah, I agree. I mean, that's something you need to do. And that's also why it's beneficial to have a pure sample, to not have everything mixing together.
Okay. And what is the throughput of the SURFE-96 SE for compound screening? Or asked in a different way, how many compounds can one screen in a day, approximately?
Andrea, please go ahead. You have more.
Yes, so I need to calculate that now in my head. So it depends, of course, on the assay design. You can one single measurement takes about 5 minutes. Yeah, so you can measure essentially 96 data points in 5 minutes. But of course, to see the effect of a compound, you need the reference measurement before. So that makes it 10 minutes for 96. And if you have all the plates already prepared, then you can multiply this essentially to get to a full 8-hour day. So it's pretty close to 5,000, I would say, if you just count reference and the compound measurement and measure all 8 hours. So I showed before the SURFE-96 SE can measure up to 10,000 data points per day, right? So you just need to take into account the controls and the reference measurements you need. And of course, the replicates if you would like to have multiple data points.
Okay. And yet another question for you, Andre, about experiments with the different potassium concentration jumps. When does the activating solution start? If, say, at 1 second, why is the peak current at 1.5 seconds or delayed rather?
Yeah. So the point is at the time point of 1 second, the valve, I mean, for the SURFE-N1 recordings, the valve switches at that time point. Then there is a delay until the solution arrives to the sensor surface. And that's about 100 milliseconds.
Okay. And do you have any idea why artifacts with the control sample are so big? How do you account for those?
We usually subtract them. So at least for the data sets I showed you, we subtracted them. And it depends on the concentration of ions. So actually, we could have measured with like, I think 10 millimolar potassium concentration jumps, and there wouldn't have been any artifacts. Then we can use the pure current. It's always up to assay design. If you want to have larger currents, then of course you might get into the range where you see significant artifacts that have to be subtracted.
Yeah. And if you change the internal pH of cells to, say, 4.2 for APC platforms, does that affect the cell health? And does it affect the duration of the recording? Either of you can answer that.
So I suppose we're not talking about live cells here. We're talking about purified lysosomes in terms.
I suppose, but yeah, that's what you can ask. Yeah.
I didn't see any differences really. Also, I found with working with 4-AP, it's washed away pretty nicely as well in comparison to other compounds, for instance. So when Andre explained that you have this initial current, then application of your compound, and then washing away the compound, 4-AP washes away pretty nicely. I'm not sure if you can add anything, Andre.
No, yeah, that's pretty.
Okay, and can you control the side of the lysosome membrane in contact with the sensor?
Not directly. As we did with the pH, if the membrane is leaky for the compound you want to essentially put into the lysosomes, then you can do it by incubation time. If not, you typically add it during sensor preparation, and during the sonication, it will equilibrate.
Okay. And one last question, is it only a single lysosome per sensor on the SURFE or a large population of lysosomes?
it's a large population. So we think it's about millions of lysosomes, and that's what brings the signal amplification compared to lysosomal patch-plan, for example.
Okay, excellent. Thank you very much. We are a little over time. We thank everyone for attending. There's just one announcement that Gemma would like to make, and then we'll be done.